quality tolerance limits as required by ICH E6R2 definitions and requirements for fit-for-purpose compliance. My name is Margot, and I'll be your X-Talks moderator for today. Today's webinar will run for approximately 60 minutes, with time for question and answer session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for future download. At this point, I'd like to thank Avoca, who helped develop the content for this presentation. The Avoca Group is a life sciences consulting firm dedicated to improving quality and compliance in the clinical trial execution process. Visit theavocagroup.com to learn more. Now I'd like to introduce our speakers for today's event. Steve Whitaker is a senior consultant with the Avoca Group and former executive director of the Avoca Quality Consortium. He is also a lead consultant for the pharmaceutical, biotech, and CRO industries, providing expertise in project management, pharmaceutical development, clinical development, outsourcing strategies, and execution plans. His experience through years of drug development leadership roles with Eli Lilly and Company, including COO for the cardiovascular platform, combined with his established professional work network across the industries, provides unique and valuable insight for organizational leaders. Steve served as executive director of the Avoca Quality Consortium from 2011 to 2018, and for 14 consecutive years on the advisory board for the annual Partnerships in Clinical Trials program, chairing the board for 2 years. In addition, he has moderated numerous quarterly clinical research consortium forums, developing strategic agendas and facilitating the exchange of leadership concepts between clinical and development executives across top-tier pharmaceutical and biotech organizations. Our second speaker for today is Jay Turpin. Jay Turpin is a senior consultant with the Avoca Group. He has been in the pharmaceutical industry for 29 years and has held various leadership roles. Jay has a wealth of experience in GMP, GLP, and GCP environments with significant real-life experience in developing numerous new drug candidates. His pharma experience started in GMP of small molecule and large molecule active pharmaceutical ingredients. Jay has been involved in leading multiple clinical teams responsible for the clinical development and NDA preparation for multiple molecules in osteoporosis, diabetes, cardiovascular, neuroscience, and immunology. Jay has also led transformation efforts for his company's CLO organization that affected processes, systems, job definitions and descriptions, and organizational leadership to meet both GLP and GCP requirements. Most recently, Jay was a senior director for the clinical project management, where he was responsible for the clinical development for Alzheimer's disease, autoimmune disease, men's health, pain, and other neuroscience indications. This CPM organization, under Jay's leadership, successfully completed multiple NDA submissions, regulatory agency inspections, and received regulatory approvals for small molecule and biologic products demonstrating thorough GCP compliance. Jay retired from Eli Lilly and Co. at the end of 2017, where he was Sir Director of Clinical Project Management and Biomedics. And without further ado, I'd like to hand the presentation over to our speakers for today. You may begin whenever you are ready, Stephen.
Okay, so thank you very much, Margot, for those introductions. What we would like to do today, Jay and I will lead you through some concepts. But before we do that, just to give you a quick framing on where the Avoca Group and the Quality Consortium provides that expertise and knowledge for the area that we will be speaking to, we would just like to make you aware that the Avoca Group consists of 3 components. It's an industry quality consortium. There's in-depth consulting and research services, and we do qualification expertise for providers related to clinical development and clinical operations. We have a positive impact on clinical trials. Our mission is to increase quality and ensure compliance across the industry and the stakeholder groups that deliver on clinical programs. The Avoca Quality Consortium in particular has the privilege of having more than 100 member companies that represent sponsors, CROs, and other service provider groups and organizations that allow us to give us the breadth of quality across the requirements that are needed to have both regulatory requirements met as well as overall quality for good interpretation of data and as well as patient safety. We have more than 400 guidelines, tools, and leading practices that come from that. And this slide just simply shows you the membership of that consortium. So it represents large, small, medium-sized companies from each of those components and groups, which gives us a breadth of leading practices that are fit and suitable for fit-for-purpose approaches. So as I approach today's session, our agenda will focus on the following topics. We look to have a clarity around the regulatory expectations on quality tolerance limits in particular, and that is largely based on the ICH E6 Revision 2 requirements. We will focus some clarification on the definitions around the quality tolerance limits, which has provided some confusion within the industry, so we hope to clarify that today. Based on that, then, what are the parameters that warrant QTLs? What's effective management of risk relative to those indicators in the QTLs tied to those? And what would be fit for purpose concepts and approaches for applying those across different phases of trials and different sponsor types? So to start off, we would like to make sure we are clear on the regulatory expectations for ICH E6 R2. So why should we care about quality tolerance limits? Again, the driver is the new guidelines, which became effective or were published in November of 2016, and regulatory agencies such as the EMA began holding sponsor companies and investigative sites and providers to those requirements as early as the summer of 2017. And other regulatory bodies began to hold organizations accountable through the course of the next year or so, such that 2018 was clearly the first full calendar year where responsibilities are being held or accountabilities are being held for member companies. For quality tolerance limits, QTLs and quality tolerance limits as a phrase or terminology does have historical precedence. It's been required for GMP activities, and it infers limits whereby significant actions must be taken. So QTLs are not just thresholds or limits whereby you may choose to do something. They are predefined, which we'll talk about, and they are points at which actions are expected to be taken. In the GMP setting, it typically is to ensure that manufactured product achieves quality and usability limits. Now that regulators are using the same terminology for GCP clinical studies, what is truly expected in this space? So if we look closely at the ICH E6 R2, I'm going to focus on section 5.0.4. Now, prior to this section, it does indicate that risk plans and quality management systems must be pre-documented at the beginning of a trial when the protocol is being developed. So with that understanding that the QTLs must be predefined at that study start level when protocols are defined, we're going to look closely then at what section 5.0.4 indicates. So if I focus on the wording, it says the sponsor should decide which risks to reduce and which to accept. The approach used to reduce risk to an acceptable level should be proportionate to that significance of that risk. This is where the phrase of quality tolerance limits comes in. Predefined, again, at the start of a study, not during the conduct of the study, but at the beginning of the study, predefined quality tolerance limits should be established. They should take into consideration the medical and statistical parameters and characteristics of the variables, and they should identify the potential systematic issues that can impact subject safety and the reliability of trial results. So again, they're focusing on subject safety and reliability of trial results. We'll come back to that because that's a fundamental, important piece of what we will be focusing on. The detection of deviations from those predefined quality tolerance limits should trigger an evaluation and action if it is needed. So it's very clear that it is intent to prompt action. If we then step further into ICH E6R2 around section 5.0.7, it also clarifies that the sponsor should describe the quality management approach implemented in the trial and summarize the important deviations from those tolerance limits in the clinical study report. So it's not sufficient just to manage it in secondary parallel documents, but ultimately it needs to be documented and recorded in the clinical study report. So at this point, I'm going to transition to Jay Turpin, who's going to give some clarification on definitions, and we will continue to give you then further details. So Jay?
Thank you, Steve. Thank you very much for the grounding that we have now on the regulatory requirements. We want to jump to a polling question at this point to see where the baseline of understanding is on these key topics. So if you will, Margot.
Perfect. So now it's time for our polling questions, where you can vote in real time by picking one of the following options. So for today's poll question, it is, I have sufficient understanding of the following terms to help my organization effectively implement them. Check all that apply. Is it metric, or the key indicators of key performance indicators, key quality indicators, or key risk indicators, parameter, quality tolerance limits, or threshold? So I'll just let the audience take a couple moments to get in their results for their voting, and then I will share those once we are done. It looks like most of everyone has voted, so I'll just wait a couple more minutes. Again, the question for today is, I have sufficient understanding of the following terms to help my organizations effectively implement them. Check all that apply. And so the results for today are 80% of you voted metric, 78% of you voted key indicators, either KPI, KQI, or KRI, 48% of you voted parameter, 41% of you voted quality tolerance limit, and 53% voted threshold. So without further ado, I'd like to hand the presentation back over to Jay. You may start whenever you're ready.
Thank you very much, Margot. So it's important as we start this conversation about QTLs to actually take a moment to determine and to define some key terminology. First of all, I want to talk to you about key risk indicators. Key risk indicators are parameters that measure operational or critical data risks for trial execution. They're intended to help support the trial in the sense that it allows leaders of clinical trials to have a sense of whether key risks are emerging and give you some foresight into what you might investigate and take forward to understand how to fix it going forward. With those key risk indicators, there are thresholds that are established. Those are limits or levels at which mitigations might be initiated. They're intended to be predefined and detect when potential issues are emerging, and the individual clinical trial leaders can actually take action to mitigate that risk and potentially bring things back into a state of control that's acceptable for the trial. Once that KRI threshold is met, again, action would be taken. Today, we're going to spend some time talking about quality tolerance limits. That term, as Steve explained, has been mentioned in ICH E6R2. Quality tolerance limits are applied to a trial parameter or a variable risk. These quality tolerance limits are intended to help clinical trial teams detect whether there are systematic trend issues that are occurring and initiate the possibility of action being taken. These risks are identified ahead of time, as Steve said, proactively at the beginning of the study, and are intended to identify what quality is adequate for a clinical trial. We'll spend a moment here explaining some of the boundaries of types of measures you might consider, but they're to actually measure quality in a clinical trial. A quality tolerance limit is a level point or value associated with a parameter. It will ultimately, if exceeded, if a value exceeds that quality tolerance limit, it should trigger an evaluation by the study team to determine whether there is a systematic issue occurring in the trial that would lead to some action on the part of the study team to resolve. Today in this webinar, we will be spending time looking at those definitions. Technically any measure could have a quality tolerance limit in a broad sense, but today when we talk about QTLs, we're speaking specifically about the requirements in ICH E6R2. So the diagram on the slide really begins to show that once a parameter is selected to apply a QTL against, we would establish this upper quality tolerance limit and then over time measure the performance of that parameter in time. It is recommended that a secondary limit be placed that actually might prompt a team to actually take action or start an investigation if a particular parameter began to exceed or rise out of a normal range, where you could proactively mitigate and investigate the issue and mitigate it and ultimately bring a particular issue back under control before exceeding an upper quality tolerance limit. The goal here is being proactive to manage the trial throughout its life, ensuring that you're achieving the quality levels that are expected in the trial. When you begin to think about the kinds of things that you could measure in a clinical trial, there's a large universe of measures that could be considered. There are measures or metrics that are used for decision-making in the trial, things like how the study's monitored, how the data management processes are being executed, safety processes. That's the broadest impact of metrics. Below that, you can measure trial procedure compliance, looking at things like the on-site monitoring performance and central oversight of a trial. So they're really process-related indicators. And then there's ultimately trial data specifications that get into the details of the trial. In most cases, quality tolerance limits are going to be established on the trial data. In most cases, you'll gather your quality tolerance limits directly from the trial data with the goal of being able to measure whether that experiment is going to be successful in measuring what it was designed to measure. Specifically, most QTLs will come from efficacy parameters, safety parameters, or other key data like inclusion-exclusion criteria. It's not to say that there may not be some critical process parameters that could be QTLs, but most likely QTLs will come directly from the trial data itself and will be measures that predict whether you're achieving the quality goals for the particular study. As you begin to define these QTLs, the first step is to, first of all, describe the parameter, spend some time actually being very clear on the definition of that parameter, what it includes, how it is actually defined for future reviewers of that data, and finally, a justification for that parameter. Why did you pick that parameter as a quality tolerance limit? Next, you'll be defining the units involved. It could be a unit like percent of a group of data, or it could be absolute values of a unit. You need to be very clear and descriptive on the unit of measure. And then what do you expect normal to be in that particular measure? And finally, a justification for that expectation. Why did you pick that measure? And why did you define it to be normal within that particular range? Explaining why you've selected that expectation. Next, you would actually then go into defining what is the absolute value of a tolerance for that QTL. What is the limit itself that you're establishing? This tolerance limit could be a one-sided tolerance limit, basically said if it exceeds a certain value, or it could be a two-sided tolerance limit where you have a minimum and maximum if you have a range you're trying to measure. Next, you would actually define a justification for that tolerance limit that you selected. Why did you pick that as your tolerance limit? Did you use past experience with this molecule from previous studies? Did you use published literature? Why did you select that particular tolerance limit? And then finally, you need to begin to proactively define mitigations or actions that you might take if this particular tolerance limit is exceeded. What actions could you take as a study team to begin to mitigate those risks? And if it continues, what is the impact of that out of tolerance situation on the overall trial? So specifically, in the next slide, we'll talk about actually what this might look like in totality. And again, these slides will be available to you. We define the parameter, we define the parameter, we justify it. Much of this is the same thing I just talked through. Very specifically on the tolerance limit itself, it should be a statistical limit of expected variance. As I mentioned, it could be a one-sided tolerance limit, or it could be a two-sided with an upper and a lower level that begins to describe how the QTL will be measured. And ultimately, the goal here is to manage risk proactively. As Steve mentioned, the ICH guidelines are focused on how can we proactively manage risk in a clinical trial. So the goal of this establishment of a QTL is to help study teams proactively monitor and manage risk that might emerge through the conduct of a study. When establishing a QTL, first of all, you need to base that determination of deciding what you're going to use as your QTL based on expert knowledge. It will need to include medical experience. Your physicians that are a part of the design of the study will obviously be a part of the discussions. Your statistician that designed the study and sample size for the study will be a part of the discussion. And the key questions will be is what risks could potentially impact the data interpretability of this study? In other words, what could go wrong that could potentially render this study as a broken study or a non-effective study? You want to predefine that in the planning phase of the study and determine what are those key measures that we should monitor throughout, and if we meet a QTL, would potentially put the study at risk. That way, you can proactively understand the status of the quality coming out of the study at the end. As you establish these QTLs, it is crucial to utilize historic data that's available. That historic data is crucial in setting appropriate QTLs. Now, obviously, that's not always possible, but to the degree you can leverage historical data, that will strengthen the validity of your QTL going forward. This data can be from previous studies on a molecule. It can be from published literature in similar studies or similar studies in a particular molecule or particular class of molecules to understand whether your study is gathering results that are consistent with that historical data. During the conduct of the study, if a QTL is exceeded, it should prompt the study team to take action. It should initiate an investigation of why that QTL has been exceeded, what's happening on the study, and ultimately it could lead to actions that the study team takes to bring the study into a better state of quality control. As Steve mentioned, the ICH guidelines also say that we need to define a summary of relevant deviations from the predefined QTLs in the clinical study report. So at the end of the study, any investigation and actions will need to be considered as you're finalizing the CSR. We also need to identify a potential secondary limit, which would give you the opportunity to take action prior to hitting a QTL. And if that's the case, you actually may be able to take actions that bring the process under control prior to hitting a QTL, and that would be true proactive management to prevent the full impact of a variance, a systematic variance that could impact the trial. We have an example of what this might look like when completed. This particular example is from my experience. It's related to measuring percent loss to follow-up patients, and it defines what a loss to follow-up patient is, how you actually define who is lost to follow-up. It defines why it's important. In this particular case, it's important because this study was a cardiovascular outcome study. It had been powered to detect a 15% difference with 80% power after treatment of 18 months or when 750 composite events were adjudicated. In this particular case, it's crucial to know the vital status of every patient at the end of the study to make sure that we can accurately assess the study outcomes and avoid missing true signals. It defines what units we're going to follow and ultimately gets to an expectation. And that expectation was that in this 10,000 patient trial, there was a goal to keep loss to follow-up patients less than 20 in that global study. Ultimately, the justification was that it had been shown that other CV outcomes trials had been able to actually monitor loss to follow-up and keep the loss to follow-up number less than 0.5%. So that led us to the goal of establishing a one-side upper limit of 0.2% at the completion of the study. In addition to that, the FDA had given advice through a review process that they expected that to be at 0.2%. So combined with industry data that had been published and based on FDA guidance, we set a goal of 0.2%. We also defined some mitigations and actions, things like spending time up front, training sites on how to minimize loss to follow-up patients. We impacted our ICF process to make sure that patients understood that we had a right to find them at the end of the study. It included defining who backup contacts were for these patients, relatives, next of kin. And we also defined up front that we might use private investigators to find their vital status at the end of the study if we could not contact them through any other means. So those were all examples of mitigation steps that were taken in our case. So hopefully that is helpful to see an example completed for you going forward. So let's talk about types of parameters that actually warrant QTLs. So as Steve mentioned, in section 5.04, the goal is to identify systematic issues that can impact subject safety or the reliability of trial results. Those are two key areas where I think you need to stay focused as you begin to think about QTLs for your study. Does this impact subject safety, or could it potentially impact the reliability of trial results? Because of this, it's really unlikely that you would focus on operational parameters unless they have the potential to impact patient safety or reliability of trial results. Recently,
this is Steve. I just want to interject to make sure that people then differentiate. This is where those operational parameters may still warrant KPIs, KQIs, or KRIs. It's just not the regulatory focus of the QTLs. So that's where we're differentiating and focusing on that specificity for the QTLs.
Thank you, Steve. That's a great clarification. Again, it's not to say that you wouldn't measure those parameters, but for the purposes of QTLs to satisfy this regulatory guideline, it's unlikely that they would be operational parameters. Now, there may be some cases where they need to be considered, but usually they will not be. Recently, in a publication through Transcelerate, these three parameters were initiated as industry-leading practice for how to establish key parameters. These three should always be considered as a starting point for your particular trial, considering, first of all, how many patients have entered the trial that did not meet one or more of the protocol entry criteria. That obviously sets up the effect of the trial, and if you have too many patients that don't meet the entry criteria, can begin to put at jeopardy the design of the trial. Secondly, considering how many subjects had premature drug discontinuation in the arms of the study. Obviously, if patients aren't on the treatment, at some point, if many of those patients stop treatment, it begins to put it in question the accuracy of the trial results of the study. And third, as mentioned in our example earlier, the number or percent of subjects as classified as lost to follow-up. If a patient leaves the study and does not complete the treatment due to over the duration of the design of the study, that obviously can reduce the accuracy and ability to measure the outcomes in that particular study at the end of the research. So these are starting points that you can consider going forward. Obviously, we recommend that each study has in the neighborhood of 3 to 5 QTLs established for the trial. These three are for consideration. There may be cases where all three of these don't make sense for your particular trial, but they're a good place to start as you're beginning to evaluate your individual trials. Now we have another polling question that really gets at who is actually leading the conversations within your organization around QTL.
Perfect. Thank you, Jay. So now I'll be launching the second poll question where you can vote in real time by clicking one of the 5 following options. The second poll question for today is, who is leading the planning and implementations of quality tolerance limits within your organization? Please select one. Is it option 1, clinical operations leader, option 2, biostatistics leader, option 3, clinical compliance officer, option 4, data management leader, or option 5, medical affairs leader? So I'll just give the audience a couple more moments to get in those polling results. And just to reiterate the question, it is who is leading the planning and implementation of the QTLs within your organization? Please select one of the following 5 options. And it looks like we've gotten our results, so I will share this poll. So the following results for who is leading the planning and implementation of the QTLs within your organization is 54% of you voted clinical operations leader, 25% of you voted clinical compliance officer, 10% of you voted data management leader, 9% of you voted biostatistics leader, with 2% voting medical affairs leader. Without further ado, I'd like to hand the presentation back to Jay. Thank you.
Thank you. It's very interesting that the preponderance of leadership comes from clinical operations, followed by clinical compliance. So thank you for that polling question. We have a third polling question that really begins to speak to when your organizations implemented QTLs. So Margot, if you could.
Perfect. All right. So when did your organization implement the use of QTLs? Now you can vote on one of the following options. So shortly effect ICH E6 R2 becomes effective, only recently, plan to the next 6 months but have not made progress, plan to the next 6 to 12 months, or no plans to implement. So I'll just allow the audience to click one of the following options. Perfect, and it looks like most of everyone has voted. So today's results for when did your organization implement the use of QTLs? The results for today are 26% of you voted shortly after ICH E6R2 became effective. 28% of you voted plan to in the next 6 to 12 months, with 20% voting only recently, another 20% voting plan in the next 6 months but have not made progress, and 5% voting no plans to implement. All right, and we may begin the presentation.
Thank you all for that feedback. So it's clear from the metrics that we are in the process of implementing QTLs across the industry. Some have done it, some are in the process of doing it now. So this obviously this webinar is timely. I'm going to turn this over to Steve, who'll speak specifically to the effective management of risk using performance indicators and QTLs. Steve?
Thank you, Jay. And again, as a reminder, the focus of the regulators on QTLs are two critical factors that are important as they review the studies that have been done by organizations, companies pursuing market authorization approvals and other aspects of clinical trials in an environment where it is important to maintain patient safety. So that's clearly one of the two factors. And then also the interpretability of the data in order to allow market authorization approvals and or label language that is appropriate. So as we look at that, again, focusing on those factors, before the trial begins, what should we really be looking at again? The trial planning stage, we want to identify the critical data or the processes that affect the study objectives, especially those related to patient safety and the interpretability of the data. It is important that the quality tolerance limits, generally speaking, the agency in the final study report does not want a massive listing of your risk plans with all the key risk indicators, key performance indicators, and so forth. They're looking to be able to interpret rapidly what you had determined to be the most significant risk factors and the parameters impacted, and what the tolerance limits you believe would affect patient safety or would affect very directly the interpretability of the results. So they expect us, generally speaking, to limit those to about 3 to 5 parameters. That prospective approach then defined the plans for those parameters and how to address the deviations from the QTLs. Again, as Jay indicated, a leading practice is to use the secondary limits to be able to then implement mitigation so that you actually don't achieve or don't hit the actual quality tolerance limit. If you hit a quality tolerance limit, it might mean that your results are not interpretable or a patient risk has become an issue. And so those are points at which serious actions should be taken. So as we look at that, at the beginning, the likely parameters are around the primary objective data, the secondary objectives data, or the patient safety data. Unlikely are the exploratory objectives or the process-focused KPIs and KQIs. We want to carefully select the very few key parameters that could jeopardize the study, as I indicated in the results, and it's the system-type errors or systematic errors. And also, proactively, those mitigation steps that could occur should be things that can be implemented to bring it back on course. Using the loss to follow-up example, what we recognize is that regulators, in the example Jay gave, a cardiovascular study, loss to follow-up subjects are needed to be classified as dead. If your endpoint, your primary endpoint is death, and you have a significant number of loss to follow-up, it could dilute the power of your study such that you are no longer able to distinguish the actual effectiveness of your treatment against a comparator arm. That is why it is such an important parameter to consider for a quality tolerance limit. Therefore, mitigation should focus on ensuring the prevention of loss to follow-up subjects. So that's an example of how you would point at the beginning of the trial. So as we look at during the trial, then you use those parameters and the secondary limits and the upper QTLs to monitor the actual measurements, the metrics that apply to that parameter over the course of time. In this example, if the metrics stay within the tolerable range, then no action is needed. It's a simple situation that you would nicely document in your clinical study report that you maintained your approach within the secondary limit. If, however, in an example is that the parameter now is showing a negative trend, such that over time you have now hit your secondary limit. If you hit that secondary limit, then you are beginning to have the risk that you get out of that tolerable range. So at this point, the potential trend would determine that you might need to implement a mitigation or a contingency and or companion metrics to be able to understand whether there are parallel factors that are at risk or that could be controlled. Again, those should have been predefined at the beginning of the study, but can then be utilized at this point to manage the trial. And then when you're in the clinical study report at the conclusion of the study, you would be able to show how you implemented mitigations and you maintained the study within the tolerance limits. So you want to evaluate the planned actions if that secondary limit and when it is reached. A third example is that you've actually hit that upper QTL. So in that case, as the time progressed, you hit the QTL, now it triggers the evaluation and an action plan. So as an example, should the study continue? Has a patient's safety risk become to a point where it is not tolerable and you should suspend the study, or some other action needs to be taken to minimize the risk to the patient? If it's related to data interpretability, are there things that you now have to document in your clinical study report about which parameters were negatively affected? So you may or may not discontinue the trial, but you then have to articulate why the study results either can be interpreted or cannot be interpreted for specific parameters, and what that means in terms of ultimately getting market authorization or label language for your trial. So you want to begin the evaluation to determine the immediate actions and what you would do at that point in time when that threshold is met. You do not want to continue to manage that trial without formally taking the actions that were preset at the beginning of the trial. At the end of the trial, then, the risk reporting is in the CSR, as I indicated. You would demonstrate how the human subject protection and data quality reliability of the trial results were maintained. You would do the summary of the QTLs, and you would summarize important deviations and the remedial actions or any implications for those results. So it's important to make sure that you realize this is a requirement from ICH E6R2, that those things must be pre-planned in the study plans and then finally documented at the end of the study, which therefore mandates very close management of those throughout the conduct of the study. So with that, let us share with you some fit-for-purpose concepts for applying those by phase or by sponsor type. So I'm going to hand it back to Jay, and Jay, you can lead us through this section.
Thank you, Steve. There is a realization that every company faces some unique challenges as they begin to think about QTLs for their particular molecules that they're in development. And what I want to spend some time just talking briefly about is that there needs to be some thoughtful consideration when selecting QTLs based on what phase of trial you're evaluating, or particularly what kind of disease areas you're focused, what kind of company you are, can have an impact on how you pick quality tolerance limits. So again, we'll focus on mostly the trial data as we go forward. So first, let's talk about phases of the trial. There should be a focus on efficacy and safety parameters. The requirements in the guidelines in ICH apply to all phases of clinical research. We've been receiving this question from many different sponsors. Does this include phase one? And the guideline does include phase one. There needs to be QTLs established regardless of the phase of trial. And again, you need to select a few key parameters. In phase 1, since you're primarily evaluating the safety of the molecule, it's likely the focus of the phase 1 QTLs will be primarily on safety, as opposed to some of the later phase development, but clearly safety is going to be front and center on a phase 1 trial. When you're establishing these points in an early phase trial, if you have similar research that you've completed in a particular disease area, they may inform the setting of the QTLs for your particular study. It will help to know the mechanism of action of the molecule and how it's metabolized, and ultimately a phase 1 team will need to think about what safety parameters are important to be monitoring as QTLs in this particular study. If there are no internal data in this particular disease area or this particular mechanism of action, it also may lead you to do some research in industry published literature of similar studies to see what other outside data might tell you and inform you as you're selecting that QTL and how you're setting that limit. Additionally, there are things like loss to follow-up that may apply to later phase trials, but may not be the most appropriate measure for some early phase trials or potentially outpatient trials. As Steve mentioned, it's extremely important in cardiovascular trials. It's also important in oncology trials to understand the vital status of a patient at the end of a study, but that may be less relevant for a phase 1 trial. So there may be alternate safety measures that you may want to consider as you establish your QTL for a phase 1 study. Another consideration is that large, medium, and small-sized companies may conduct trials in the same disease categories. And again, there should be a growing understanding of what QTLs are being used across an industry for the same disease categories. There's a real need for us to monitor peer-reviewed published articles to see what QTLs have been selected by other sponsors. And ultimately, as time marches on and we all gain experience with QTLs, we believe that an industry-wide collaboration should evolve, where we are all sharing the QTLs selected in a more open way. These are really all intended to focus on the patient safety and the efficacy for the patient. So as we think about what's good for the patient, across the industry, a broader collaboration really is, I think, a future state that we should all strive for, where we share QTLs and we drive greater and greater consistency in disease categories through that sharing. There's also some special considerations for some particular diseases, like rare diseases, where it may not make a lot of sense to measure loss to follow-up, but it may be more important to measure things like treatment compliance. This is especially true in acute treatment trials and in trials of rare diseases. And so, again, you'll have to consider your specific circumstance to try to set quality tolerance limits on the measures that make the most sense for your disease that you're studying and the experiment that you're running in your trial. As a team actually establishes their QTLs, they actually need to make sure that they're applying these across stakeholder groups. Today in the industry, when we implement clinical trials, there's a broad group of people that have to partner together to make it happen. So stakeholder groups like the sponsor, the CRO that's running the trial, the technical service providers that are supporting the trial, and investigative sites need to collaborate and need to be aware of the QTLs that are being monitored so that they're all working together to ensure the data is of the highest quality and that ultimately the study is managed in a way that proactively protects the safety of the patient and protects the integrity of the data results. So you have a study that can be interpretable and ultimately can be counted on. The quality can be counted on at the end of the study. So in summary, QTLs are established during the planning stages of the trial. This needs to be done proactively. The purpose is to identify systematic issues that are occurring during your trial that may impact subject safety or the reliability of trial results. After this QTL is established, if when monitoring that QTL it has been exceeded, it should trigger an evaluation of the deviation that has happened in that particular parameter. Ultimately, important deviations and associated with that parameter and QTL are required to be reported on in the CSR of that study, as required again by ICH E6R2, and you need to be prepared to speak to any remedial actions that have been taken in that particular deviation. So with that, we're going to pause and open it up for questions. Are there any questions?
Perfect, thank you. Yes, now it is time for our question and answer portion of the webinar. So I invite our audience to keep sending in those questions through that chat box, and I've already received some, so we'll start with those. So the first question for today is, what is the best way to link RACT critical data and processes and mitigations to key risk indicators?
So this is Steve, I can answer. So the RACT, or many of you use what we are known as RACT tools. Those are the risk assessment and categorization tools. And there is absolutely an approach to use those existing tools to support QTLs. What you would want to do, however, is you would not want your parameters that are tied to quality tolerance limits to be lost in what might be a voluminous number of overall trial managed risks throughout a program. You would want those to be distinguishable in some fashion. So whether you prioritize them, color code them, some way you want them extractable and measurable as individuals so that they maintain the most highest scrutiny. The other thing you'll want to consider is many companies, oftentimes even small pharmaceutical biotech companies, might rely on the provider, the CRO, and their use of a RACT to manage the trial. At the same time, you'll realize that the CRO will tend to focus most on the operational risks. And so if the sponsor delegates it to a provider to do the RACT, then the provider predominantly is the one most impacted by the regulations of ICH E6R2 and the patient safety or data interpretability risks, because those affect your ability to complete a study and to ultimately get market authorization approval and label language. So I would highly recommend that the QTL factors, the parameters that affect QTL, be closely scrutinized and managed by the sponsor in partnership with providers.
Steve, I might add that in our consulting practice, we've had several questions related to this. And it is industry best practice when that RACT tool is completed, that actually be done with all the stakeholders having a voice in that process and not just the CRO completing the RACT tool. It's really best practice to have members of the sponsor team participate in that risk assessment so that you can move beyond just operational risks and also consider other data risks that are ultimately the source of most of the QTLs that we would identify.
Great, thank you for that response. Our next question is, can you please give an example of a QTL?
We have given 3 examples in the presentation, like the loss to follow-up. Others that I would, so those, rather than repeating the 3 that are in the slide deck, people can go back and review those since the slide deck will be available. Another concept to think about would be the following, and that is, if I were to conduct a study, and if I'm doing, let's say, an oncology study, there are data not just for treatment clarification, but even disease-related biochemical measurements. So you typically would do chemical screens, things of that nature, and you know the parameters whereby an oncology patient through normal treatment might become so ill or to a point where it is not tolerable from that patient perspective. So those data, whether they're historical data or disease-related data, should also be part of what you consider to determine whether that should be a parameter where your treatment could impact it in some way because of the mechanism of action by your treatment, whereby you should set a predefined parameter and tolerance limit that you're going to closely scrutinize. So again, if you know the mechanism of action might affect that biochemical parameter, that should definitely be one to consider for a QTL.
Thanks. I was thinking about even in phase one, when you're looking at safety, if you have a small molecule that you know is metabolized in the liver, you may choose markers of metabolites and have some thresholds that are really focused on patient safety, and those might ultimately be the right QTL for an early phase one study looking at the metabolism of a new small molecule.
And that brings us to our next question, which is, do QTLs depend on available resources? If resources are unlimited, everything could be monitored.
It's a great question. I would say that QTLs are not negotiable relative to resources. It is a compliance requirement by regulations. I would say there may be some other key risk indicators, key performance indicators that are more operationally based, that if you are to, due to capacity or resource limitations, decide not to monitor something, it would be in those area spaces, not in the parameters that relate to quality tolerance limits.
Thank you for that response. And our next question, is it expected that the risk assessment and categorization tool high risk line items be monitored by KRIs and their mitigation should their mitigation match?
So Jay, feel free to answer, but I would say again, so that's the important piece. You may have key risk indicators that are operationally based, and those could have high impact and high probability such that they desire and require close scrutiny, but it might be, let's say, timeline based. So as an example, it's going to be important for an organization to achieve a certain timeline to be able to maybe meet or beat a competitor product in development and or to meet certain financial commitments to Wall Street and things of that nature. So those can be very high important racked items that would be managed, and again, you would want to prioritize those, but those generally would not be parameters for quality tolerance limits because that timeline is more of a financial implication for an organization and not tied directly to patient safety or interpretability of data. It's not to say that timelines couldn't. There could be situations where timelines do affect patient safety or data interpretability, but if it does not, those can still be managed by a RACT, just not tied to QTLs.
Great. Great, thank you for that response. Our next question is, you are referring to QTLs mainly related to data interpretability. What about the patient safety aspects?
Yes, I'll take that one. And again, I think each study needs to evaluate what are the risks associated with that particular experiment. There are clear needs to monitor certain patient safety parameters throughout the life of a trial. In particular, I think this is going to be more predominant even in early phase trials as you're beginning to characterize the metabolism of the molecule. Safety is going to be, highly sensitive in the early stages of development. And in the confirmatory studies, you obviously will continue to monitor key parameters. So again, any particular areas of metabolism that could add risk to the patient might be candidates for a QTL. It could be related to, again, liver function, enzymes. If it's metabolized by the liver, it could be parameters of performance in the kidney, depending on how it's excreted. Those are considerations each study team should consider as they think about possible safety parameters that should be considered as possible QTLs.
Well, thank you very much for those answers. Unfortunately, we've reached the end of our question and answer period. If we weren't able to attend your questions, you may now direct them to the email showing up on the screen. Avoca may try to follow up with you in writing in the next 3 to 5 days. And now I would like to introduce the upcoming Avoca webinars and conferences. The Avoca Group will be presenting on several topics at the upcoming Crown Congress and TMF Summit, which are both co-located at the Hilton Orlando Lake Buena Vista, January 22nd to the 24th. If you'll be attending either of these events, please join Avoca for one of their sessions. You can save 50% off on your registration by entering the discount code Avoca during registration. As a precursor to Avoca's presentation, EXL's upcoming third European Clinical Quality Oversight Forum, February 25th to the 26th in London, Avoca will be giving a webinar on January 29th. And as a precursor to Avoca's presentation at EXL's 10th Proactive GCP Compliance Conference, March 18th to the 20th, Philadelphia, Avoca will be giving a webinar on February 13th. Thank you for everyone participating in today's conference. You will be receiving a follow-up email from X-Talks with access to the recorded archive for this event. A survey window will be popping up on your screen, and your participation is appreciated as it will help us improve our webinars. You can share this webinar on LinkedIn by the following link that's been set out in the chat box, thanks to Steve Whitaker and Jay Turpin. We hope you have found this conference informative, and have a great day, everyone.